Welcome to Efiwe, excellence without stress.
So good day everyone and welcome to another Efiwe Jam Biology tutorial, Excellence Without Stress. So today we'll be talking about cell and its environment. We've talked about cell in a previous video, and today we're going to be talking about the cell in relation to its environment. So we're going to talk about things like osmosis, diffusion, plasmolysis, hemolysis, and other things like that. First of all, we'll start with introduction. So in biology, the study of the cell and its environment is crucial to understanding how cells survive and function. So in the environment they stay, how do they survive? How do they function in that environment? So one key aspect of this topic is the movement of substances across cell membrane. So we also talk about the cell membrane of a cell, which can occur through processes such as what? Osmosis, diffusion, active process, and passive transport. So we're going to talk about active transport and passive transport. But one important concept that's related to this topic that you have to have an idea of is what we call semi-permeable membrane or semi-permeable membrane. So which allows what certain molecules to pass through while preventing others from crossing. From the name semi, it's like half. So some things will pass through, but other things cannot pass through. So this membrane is crucial to maintaining what the internal environment of the cell and regulating the movement of substances in and out. So it helps some substances enter, helps some leave, and it doesn't allow all. So it's very important in maintaining. It doesn't just allow anything enter into the cell. So things that are useful to the cell, it allows it enter. Things that are useful to the cell, it doesn't let it leave. So it's things that the cell does not need that it lets leave. So we call it what a semi-permeable or a semi-permeable membrane. So first of all, we'll talk about diffusion. So when we discuss the movement of substances, it is important to consider the concentration gradient. So what do we mean by concentration gradient? It's the difference in concentration of a substance between inside and outside the cell. So are they more inside the cell? Are they more outside the cell? That's what we'll talk about. What concentration gradient? Is there more inside the cell? Is there more outside the cell? Just the difference. If they are equal, there is no concentration gradient. There have to be difference in concentration between inside the cell and outside the cell for them to be what a concentration gradient. So when the concentration gradient is high, molecules will move from what? From areas of high concentration to areas of what? Low concentration. And this is what we call diffusion. So diffusion is the what? When there's a concentration gradient, that means difference in concentration between inside the cell and outside the cell. Things will tend to move from what? High concentration to low concentration. So what diffusion? The diffusion is the net movement of molecules or atoms from a region of what? High concentration to a region of what? Low concentration. So this is a passive process. And we'll see what it means by passive process very soon. So diffusion is the passive movement of particles from a what? Area of higher concentration to an area of what? Lower concentration. So this process is essential for what? The exchange of gases and nutrients in the cell. It's very important for what? Exchanging nutrients and gases. So things like oxygen and CO2. If you watch many of our videos, videos on transport, videos on respiration, we'll talk about things diffusion, diffusing in and diffusing out. So this is what we mean by diffusion. If there's more oxygen in the lungs than in the blood, so oxygen will move from the lungs to the blood. So it's diffusion. If there's more CO2 in the blood than the lungs, so what? CO2 move from the what? From the blood into the lungs. So the movement from the higher concentration to the lower concentration, we call it what? Diffusion. So, but what active transport? So, in addition to simple diffusion, we just talked about what simple diffusion. So, in addition to that simple diffusion, cells they will also rely on processes like active transport to move substance against the concentration gradient. That means things now are moving from what low to high. So normal diffusion, things move from what high to low. That's normal, passive, no energy. But when you're now moving it against that concentration gradient, that means from what low to high. So we call it what active transport. So this requires energy in the form of ATP. I've told you before in the previous video that what the currency for energy in the cell is what ATP, adenosine triphosphate. So it allows the cell to maintain higher concentration of certain molecules on one side than the other. Let's say the plant need or the cell need maybe glucose, and there's already more glucose inside the cell than outside the cell. On a normal note, it's not the glucose is meant to flow from inside the cell to outside the cell because we already said that division is movement from higher to lower. But there's higher inside the cell, but the cell even needs more glucose. So we need to move this glucose from the smaller side, which is outside the cell, into the cell because the cell needs to accumulate glucose. So that's what we call what active transport. So we need active transport or something like that. So what exactly is active transport? So it's the movement of ions or molecules across the cell membrane into a region of higher concentration and is usually assisted by what enzyme and it requires what energy. So active transport is the movement of molecules across a membrane against their concentration gradient. Please take this is a very important point against their concentration gradient and usually requires what ATP. So this process is crucial for transporting ions or molecules against their natural flow. So it's meant to flow like this, but no, we want it to go the other way. So we push it. So that what we call what active transport. From the name active, it requires energy. So if there's active transport, there will be what? Passive transport. Because the opposite of active is what? Passive. So positive transport is the movement of ions or other atomic or molecular substances across cell membrane without energy input. You don't need energy. Passive, it just goes normally. The positive transport involves the movement of molecules across the membrane without the need for energy input. So simple diffusion that we've talked about, and also we have what we call facilitated diffusion. It's still diffusion, but it just helps the diffusion. So those are examples of passive transport. So we have simple diffusion, that's just when diffusion occurs on its own. Then we have what? Facilitated diffusion. That's when diffusion is assisted, but it's still diffusion. So we call it passive process. You don't need energy. So take note of that. So we have semi-permeable membrane. So a semi-permeable membrane, what? It's also termed what we call it selectively permeable membrane, or we call it partially permeable membrane, or a what? A differentially permeable membrane. So these are the other names you might see. So please take note. You might see any of these names. So a type of biological or synthetic polymeric membrane that allows certain molecules or ions to pass through by diffusion. I've said it before. Certain things pass, certain things don't. It doesn't allow all things. If it's freely permeable, anything can pass through. But once it's semi-permeable, some things can pass through, some things might not pass through. So the semi-permeable membrane allows the passage of certain molecules or ions while restricting the movement of others. Allows certain to pass, allows it to not pass. From the name semi-permeable. So cell membranes, they are examples of semi-permeable membranes. Cell membrane I've talked about, they are what? Examples of what? Semi-permeable membranes. So when the concentration of a substance is higher outside of the cell than inside, the cell will either gain or lose water through molecules. Through osmosis. So depending on the tonicity of the solution. So we'll talk about what osmosis means very soon. A solution that is hypotonic, please take note. Hypo means lower. Hypotonic. So that solution is hypotonic. So that means what? The solution will have a lower concentration of solute than the cell. That means what? Inevitably, that means what? The cell will have higher than the what? Solution. So causing the cell to what? Gain water. So water will move from where is high to where is low. So if there are more solutes inside the cell, that means what? Less water inside the cell. And if there are less water inside the cell, that means there are what? More water outside the cell. And water, this will move from what? Higher to lower. So this will move from outside the cell into the cell and lead to what? To gut pressure. So, but on the other hand, hypertonic solution, the solute concentration of this the cell. Sorry, for an hypertonic solution, the solution has more solute. And if you have more solute, that means you have what? Less water. So if there's less water outside of solution, that means what? Inside the cell, there'll be what? There'll be what? More water. And water will move from where is high to where is low. That means water will move from inside the cell to outside the cell. So causing the cell to lose water and potentially lead to what? Plasmolysis. So from the name iso, one here, iso means equal. So an isotonic solution, where the concentration of solute is equal inside and outside the cell, causes no net movement of water. So inside and outside the cell, they are the same. So things will not move. So take note, please, even though you don't gain anything from this lecture, please take note of all these things. A hypotonic solution has less solute and more water than another solution. Hypotonic from the name, it has what? Less solute. So if there's less solute, there'll be more water. It's just opposite. If there's more water, there'll be less solute. But for an hyper, when somebody is hyper, that means too much. That means it has more solute. And if there's many solute, there'll be less water. But for an isotonic solution, it contains what? Equal concentration of solute and water. So hypo less, hyper more, iso the same. So what exactly is osmosis? We talk about osmosis. What is osmosis? So osmosis is the spontaneous net movement of solvent molecules or water. We say water is a universal solvent. Water is a solvent. So most essentially water through a what? A semi-permeable membrane. So now this is the difference between osmosis and diffusion. For osmosis, there must be what? Semi-permeable membrane. It's compulsory. It's a must. It's a must. It's a must. That's why I put it in bold. So through a semi-permeable membrane, what? Into a region of higher concentration. So in the direction that tends to what? Equalize the solution between the two. At the end of the day, there should be equal solute in the two. So osmosis is the movement of water molecules across the world, a selectively permeable membrane or a semi-permeable membrane from an area of what? Low concentration to an area of what? Higher concentration. That's of solute. But if it's water, the water will move from what? High water concentration to low water concentration. I don't know if you're getting it. If it's solute, it's the opposite. No, solute is the opposite of water. For osmosis, water will move from what? Where is high to where is low. And if there's high amount of water, there's low amount of solute. If there's low amount of water, there's high amount of solute. So please take note. If you're talking about water, it will be different from when you're talking about solute. So please take note. So it plays a crucial role in what? Maintaining the cell shape and function. So if the cell are taking in more water, it will maintain the shape. It will be solid. You get. So that's what it means by that. Now talk about more about hypotonic, hypertonic solution because this is the most important part. Most of your exam questions they're going to ask you is going to come from here. Hypotonic, hypertonic. When the cell is placed there, what happens? When the cell is placed there, what happens? From the name hypotonic solution, it has lower solute concentration compared to the cell cytoplasm. So the cell, inside the cell, we have what? More solute concentration than outside the cell. So when we talk about solution is outside the cell. So if you say hypotonic solution, that means the solution which is outside the cell has what? Lower solute concentration. And if it has lower solute concentration, it will have what? More water concentration. So cell plays in hypotonic solution what? With gain water. Because water will move from what? Where is high to where is low. It's very easy. Listen, I don't cram it. I can still remember it. They taught me how many years ago. I can still remember it. Just easy. Just calm down, analyze it. So from the name hypotonic solution, we're talking about the solution, not the cell. Outside the environment. So if it's hypo, that means going to have what? Lower solute concentration. Hypotonic solution. So it's going to have what? Lower solute concentration. And if solute concentration is low, the water concentration will be what? High. And water move from what? High to low. So the water will move from outside the cell into the cell. So and if it continues entering through the cell, at the end of the day, the cell will burst. So we call it lysis. Because if it continues taking water, there's a particular point it cannot take more than more to burst. So we call it what? Lysis. So let's talk about what hypertonic solution. So hypertonic solution has what? From the name hypertonic solution, it have what? Higher solute concentration than the cell's cytoplasm. So if the solution has what? Higher solute, that means it's going to have what? Less water. So if the outside of the cell is having less water, that means inside the cell has what? More water. So water will move from what? Inside the cell, outside the cell. So cells in a hypertonic solution what? Lose water. You lead to what? Shrinkage, now become smaller, or what we call it what? Plasmolysis. So it's very easy to understand. You just have to calm down and understand. You don't have to cram it. Just calm down and understand it. So an acetonic solution has the same solute concentration as the cell, so it's equal, iso, equal. So cells as a solution will maintain their shape and size. Nothing will happen. No water will move, no water will leave, no water is entering. It's the same. So we call it a what? Isotonic solution. So what is happening in plasmolysis? So plasmolysis is the process in which what? Cell lose water in what? In a hypertonic solution. We talked about this before. So just calm down and think about it. So the reverse process is called, we call it what? Cytolysis. So it can occur if the cell is placed in what? Hypotonic solution. So I will not even try to explain it again. Just calm down and think about what I've explained before. And it results in a what? Higher external osmotic pressure and a net flow of water into the cell. So if you don't understand it, you can watch the video again, but calm down and think about it. It's very easy. Hypertonic solution. That means the solution has what? More solute than inside the cell. And it has what? More solute. That means it has what? Less water. So if outside the cell has what? Less water, that means inside the cell has what? More water. So where is water going to move? Very easy. So plasmolysis is the process in which a cell loses water in a hypertonic environment and it causes the cell membrane to contract away from the cell wall. So the cell membrane will contract away from the cell wall. So this occurs when the external solution has what? Higher solute concentration than what? The cells. Just calm down and think about it. It's actually very, very easy. So these are questions that when you see it, you just laugh through and you just get it. You don't have to spend much time here. So what about turgidity? So this is the state of what? Being turgid or swollen. Turgid. Okay, turgid. So especially due to what? High fluid content. So it is important the plant cell as it allows them to remain rigid. So if you hold them, they are rigid because they don't have support like the way we do. They don't have bones. Like me, I can stand because of my bones. But plants they can't do that. So they need to be turgid in order to maintain their shape. So and hold their shape. So turgidity is the state of a cell when it's swollen due to what? Water inflow. That means water has flowed into the cell. So the cell will become making them firm and what? Rigid. So it happens when the cell is placed what? In an hypotonic solution. So where the external solute concentration is lower than the cell's cytoplasm. Just sit down and think about it. It's actually very easy to understand. So what about hemolysis? So hemolysis is the rupture or destruction of what? Red blood cell. When you see lysis means breaking. So hemo is red blood, hemoglobin. Anything hemo is what? Blood. So it's the destruction of what? Red blood cell. And it occurs when the blood is placed in what? Hypotonic solution. And it causes water to move into the cell. So after water has moved into the cell, it bursts. But plants, they don't burst easily like that. They become turgid because of their cell wall. This is very important point you have to take note of. So hemolysis is the rupture or extraction of red blood cell and releases what hemoglobin into the what surrounding fluid. So it typically occurs in what hypotonic solution, causing water to enter the cell and leads to what swelling and what eventually bursting. So finally, it is worth noting that only water moves through the cell membrane. So discussed within this context, hemolysis refers to the destruction of red blood cell that can occur when in place what hypotonic solution. So all these processes of water moving in, water moving out, they are fundamental to the understanding of cell and its environment, and they play a crucial role in maintaining the cell's internal balance. So it maintains things where the cell is balanced inside and outside of the cell. So these processes are very, very important. So that will bring us to the end of today's lecture. So thank you very much for sticking with me to the very end, and I'll see you in the next lecture. And please, please, and please make sure you download the app. Thank you.